Hi, class. In this recording, we're going to talk about a lot of different things. It's kind of a mishmash of topics. We're going to focus on your field of focus, your peripheral vision. We're going to focus on stereoscopic vision. And we're also going to focus on senescence, or the loss of function of our special senses as we age. So let's dive into it. When we think of the duplicity theory of vision, this explains why we have both rods and cones in our eyes. So the rods, just to review, rods are for low light environments, and there's one kind of rod. Cones are for high light environment, and these are going to be our color vision. Think C for color, C for cone. And there are three primary kinds of cones, and we use those rods for our night vision, and they're good for black and white vision. As we look at our receptors, the rods and cones, a single kind of receptor can't be sensitive and precise or have high resolution. So when we think of the rods are super sensitive receptors. When we think of our cones are our super precise or super receptors that give us, excuse me, blah, 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 blah. Cat has my tongue. The cones are the receptors that give us the high resolution or the very precise vision. So as we look at these cells, it's worth emphasizing that it takes one kind of cell and neural circuit for sensitive night vision, aka the rods. And it takes a different kind of cell, the cones, for that high resolution daytime vision. So rods react in dim light. And when we think of rods, there's lots of neuronal convergence. So when we say neuronal convergence, we're talking about how many rods will ultimately make it to one ganglion cell. So here we have lots of rods. We have multiple rods converging with one bipolar cell. And then we have multiple bipolar cells all converging with one ganglion cell. So in this figure, we have 1, 2, 3, 4, 5. Looks like I'm estimating about 15 rods all converging to the same ganglion cell. And this figure simplifies things. In our retinas, we can have as many as 600 rods converge to the one bipolar cell, and then those multiple bipolar cells all converging to one ganglion cell. What does this mean? This means that we can have it's not uncommon to have well over 1,000 rods converging at one ganglion cell. So this means that we have a high degree of spatial summation. One ganglion cell is going to receive information from 1 square millimeter of our retina. And this means we have low resolution and we get a very coarse image. In other words, when it comes to night vision, we don't have very good night vision. In the dark, we don't have high resolution. I'm thinking just last night when I woke up in the middle of the night to go to the bathroom, I walked out into the hallway. There was a vacuum cleaner in the corner of my hallway, but I swear it looked like the possessed doll Chucky from that classic horror movie, and it freaked me out a little bit. And I had to flick on the light to calm my nerves down. And that's because our night vision has coarse resolution, and it's really hard for us to see well in low light environments. The edges of our retina, aka our peripheral vision, have very widely spaced rod cells that also are going to act as motion detectors. What does this mean? This means that in our peripheral vision, we are going to be preferentially attracted to things that move. Why do we have this? Why don't we have a high concentration of rod cells and cones over the entire retina? It has to do with processing. Processing all of these signals from the rods and cones eats up a lot of brain matter. It takes a lot of neurons in our brains to process this image. And because we don't want to I'm saying from an adaptation perspective, it's not advantageous for us as an organism to have an occipital lobe that's 4 or 5 times larger than it currently is, so that can process high resolution images from across our entire retina. So we split the difference and we have high resolution images at our field of focus. But for our peripheral vision, we just focus on the really important stuff, which is things that are moving that might be sneaking up on us from the side. So within our excuse me, I'm having brain up here. Within our retina, there's the macula lutea, and in the center of the macula lutea, we have our fovea centralis. I want to emphasize here, look at this. There are only cones. there are no rods. There is also no neuronal convergence. We have a 1-to-1 ratio of cone to bipolar to ganglion cells. This means, think of this like like motor units from our skeletal muscle system or our muscular system. This means that we have the equivalent of very small motor units. We have very precise controls. Each cell, each cone has a private line to the brain. This means that we have very high resolution image processing when we look directly at something and take that light from whatever we're looking at and focus the light on the macula lutea and fovea centralis of our eyes. Let's talk about something here. Because there are no rods in the fovea centralis, the center of our field of vision is terrible for night vision. You may have noticed this if you ever looked at stars in the middle of the night. So the next time you have a chance, go outside when it's not cloudy, look up at some stars. And then for me, the classic example of this is the Pleiades cluster near the Orion constellation. And I remember as a kid, when I would try to look directly at the Pleiades cluster, I would look directly at it and it would almost disappear. Most of the stars I can no longer see within that cluster. But then I would direct my field of vision off to the side, and all of a sudden, in my peripheral vision, the Pleiades cluster would light up, and I could see all these extra stars in it. What's happening here is that in a low light environment, like stargazing, for example, when you look directly at a star, you don't have any cone or rods to receive the dim light from the star. And a lot of times that star disappears. But when you turn your field of vision away and you put your favorite star, the one you want to see in your peripheral vision, all of a sudden you can see it. Or, for example, when I walked outside my bedroom last night and there was the possessed vacuum cleaner that I thought was a doll from a horror movie. When I looked directly at it, it looked like the Chucky doll. But when I turned my peripheral vision on it, I looked off to the side, I could tell it was a vacuum cleaner, and my pulse rate went down a little bit. As we look at this spatial summation, or the fact that we don't have any rods in our fovea centralis, we have less sensitivity to dim light. I'm going to throw out there we have almost no sensitivity to dim light relative to our peripheral vision. Now, as we think of human beings, we humans are a fancy kind of primate, Homo sapiens, and we have well-developed color vision. If we look at nocturnal vertebrates, organisms that are typically active only at night, are only going to have rods because there's no sense in them having highly sensitive photoreceptors when there's not enough, or highly color photoreceptors when there's not enough light for those nocturnal animals to see in color. So nocturnal animals typically will only have rods in their retina. But since we are diurnal, we're typically awake during the daytime, it makes sense for us to have cones that can pick up color vision. And as we look at cones, we as human beings have three kinds of cones in our eyes. We have 1, 2, 3. The S, M, and L stands for short wavelength, medium wavelength, and long wavelength. We as human beings have cones that specialize, that have peak absorption spectra at 420 nanometers, 531 nanometers, and 558 nanometers. And I'm going to clear the screen for us for a second here. For any of you that have spent any amount of time doing spectroscopy or summation of graphs, you know that if we add up our color vision, we'd have a peak here. And then at this point right here in this highlight environment, we'd have to add the green, the peak value of the green, plus the peak value from the M cone, or the peak value from the S cone and the peak value from the M cone. So we actually have better sensitivity. If we add these two peaks together, it actually would be about right up here. So we could have a peak absorption right here in highlight environments. But look at this M cone and this L cone. These M and L cones have pretty close peak absorptions. And if we add them together, what we find is that, let me clear the screen. Let's go with blue right here. What we find is that the M cone and L cone overlap so much that at this wavelength right here, we have very, very high rates of absorption. This is the frequency or the wavelength of the most easily seen color for us, which happens to be the safety green color. The safety green is the most easily absorbed color by our retina based on the M and L cones peak frequency of absorption. And this means that it's the easiest color for us to see. So when we perceive different colors, like think of the Roy G. Biv, the colors of the rainbow, we're going to have a mixture of S, M, and L cone stimulation mixed and matched to give us different colors that we're perceiving. So to see green, we're going to have activation of both the M and L cones. To see violet, we're going to have only S cones activated. To see red, we're going to have only the L cones activated. So based on these different mixtures of activation, we can perceive that as different colors. Let's move on to stereoscopic vision. When we think of stereoscopic vision, I want you to think of having binocular vision or perception vision, the ability to judge distances. As you look at different objects that are different distances away from your eyes, those objects are going to have different varying amounts of deflection. Near objects are going to be deflected more towards the lateral margins of our retina. Very distant objects are going to be deflected slightly towards the medial margin of our retina. And then we can have our fixation point, our frame of reference. So what does this mean? Whenever we're looking at something in space and we're fixating on it, anything closer than what we're looking at will be deflected to the lateral margins of our retina. Anything farther than what we're looking at will be deflected towards the medial margins of our retina. And this misalignment of those objects within our peripheral vision is going to give us double vision. But the fixation point is going to allow, when we look at something or fixate on something, we can use the angle that we need to move our eyes as a way to automatically calculate how far away something is from us. We do calculus automatically in our brains. And I'm not talking about like calculus in terms of writing numbers on a piece of paper and spitting out an integral or a derivative. I'm talking about the ability to look at something and intuitively know how far away it is, and also to intuitively predict the curve of an arc and where something is going to be so we can catch a ball, for instance. Something else that we do automatically in our brains, subconsciously, is we do trigonometry without even thinking about it. So as we look at stereoscopic vision in our depth perception, this is going to involve the angle that we tilt our eyes at, and it's also going to involve subconscious trigonometry and calculus being performed within our brains. Pretty cool. Now, if you think of an organism like a squirrel, and I have lots of squirrels in my backyard. Squirrels, they are prey. Prey organisms typically are going to have their eyeballs on the left and right sides of their brains, and those eyeballs typically don't have overlapping fields of view. Prey organisms typically don't have good stereoscopic vision. Hunter organisms, like human beings, we typically have our eyeballs on the same side of our face or the same side of your skull so that you can have overlapping fields of vision, so you can have depth perception and better judge how far your prey is away from you, so you're a better hunter. Oh, wrong button. There we go. So let's talk about senescence of the system, the special senses, or loss of function of our special senses. And typically for vision, one of the first things that occurs as we get older is we lose flexibility of our lenses. We have presbyopia. Oh my Lanta, that's a tongue twister. Effectively, this means that as we get older, we lose the ability to focus on things that are really, really close to our face. And we need to start wearing cheater glasses to read. Something else that happens as we get older is that the lens of our eye becomes cloudy. A cloudy lens, which is shown here, this is a very cloudy lens, is known as a cataract. Something else that happens as we age is that we lose our night vision. We have fewer and fewer rods in the back of our eyes. We lose those rods. And also, the vitreous humor, vitreous body in the back of our eye becomes less transparent. So it's low light environments we have a harder time seeing. We need to have brighter lights in order to see. Also, the pupil dilators start to atrophy. This means that it's harder for us to open up our pupil and let more light in. And the enzymatic reactions that restore the rhodopsin are also slower. So think of this in terms of driving on a country road at night. One of the reasons why you turn off your high beams isn't just to be nice to the other people that are driving, because you don't want to blast them with your high beams while you're driving down the road at night. You turn off your high beams so that you don't bleach their photoreceptors, and so that they're better able to see your car and not hit your car. But then again, on the flip side, when there is no oncoming traffic, you turn on your high beams so that you have more light and you're better able to see things like those deer that are trying to jump out of the cornfield and sacrifice themselves in front of your car. Something else that happens is that if we age, we also have an increased risk of glaucoma on our eyes that can be quite painful for many individuals. As we think of hearing, as we age, the tympanic membrane and the ossicles, the malleus, incus, and stapes, tend to stiffen. And the stiffening of the membrane and auditory ossicles, means that we have a harder time taking a sound wave and transmitting it to the inner ear to vibrate those hair cells that we use to make the action potentials. Also, the hair cells within the inner ear start to die. As those hair cells start to die off, their axons, which constitute the auditory nerve fibers, will also die. We also have neurons in the semicircular canals and the succuli, or the vestibule of the inner ear, and those neurons tend to die off as well. As we lose vestibular neurons, we have a harder time maintaining our sense of orientation, and we oftentimes become dizzy, or we develop vertigo. Vertigo is more common in elderly than it is in the young because of the death of vestibular neurons. Also, as we think of taste and smell, and I know we didn't spend a lot of time in this particular class talking about taste and smell, but we have blunted receptors as we age. What does this mean? This means that as we age, we're able to eat stronger and stronger flavors. When we're young, many kids don't like coffee, beer, olives, those, or sauerkraut, those foods that have really strong flavors. I know for me in particular, I hated mustard or Grey Poupon when I was a young kid. Now I think it's fantastic tasting because my taste receptors have been so blunted over the years. And smell is another one. We start to lose our sense of smell as we age. This is one of the reasons why you typically will find that older men will have lots of cologne or aftershave on. To them, it smells very normal, but to younger individuals, it smells very overpowering. Or I'm thinking of some time I spent working with some kids at a nursery. I could smell the poopy diapers, no problem. But some of the more geriatric individuals I was working with couldn't smell the dirty diapers at all. They were happily oblivious to that nasty odor that was emanating from a couple kids' pants. As we look at hearing, or sense of hearing as we age, young individuals typically are going to have a very wide range of hearing. And then geriatric or elderly individuals will have a narrower range of hearing. And I want to emphasize something here. As we look at our range of hearing, the low frequency noises, generally speaking, remain the same. So whether we're young or geriatric or in between, we typically can hear low frequency noises pretty well. But as we age, we lose the ability to hear high frequency noises. So when we're working with geriatric individuals and you're talking to somebody who's hard of hearing, it might be helpful, particularly if you have a high-pitched or high-frequency or feminine-sounding voice, to talk in your man voice. Lower the frequency of your speech so it's easier for somebody to hear what you're saying. I know we went a little long in this recording, but it was a really complicated video with 3 rather complicated topics. If you have any questions or comments, please feel free to post in the class discussion board or to shoot me an email. And as always, happy studies.